So the digestion and absorption in the GI tract occurs through the breakdown of the ingested foods into their chemical building blocks. And these chemical building blocks are the individual monomers, which are the units small enough for absorption. So what we ingest are macromolecules, large giant molecules that need to be broken down by monomers via physical means or by enzymes. And those enzymes, they carry out hydrolysis, which is where water is added to break a chemical bond so that the monomers are present and they can be absorbed. So the mechanisms of absorption is the process of moving the substances from the lumen of the gut, that's the small intestine or the GI tract, into the body. And remember, the small intestine is the main location where absorption actually takes place. So here we can see the route of absorption through an enterocyte. That's just the name for the individual cell that's lining the small intestine. And once it's absorbed, then it can enter into the bloodstream where it goes to the liver for processing. Remember, via the mesenteric veins that travel to the portal vein, the hepatic portal vein system. So the first main nutrient which is broken down in our body is carbohydrates. And they need to be, the starch, the larger molecules, they need to be broken down into smaller molecules, monosaccharides, before they can be absorbed. And this process begins with salivary amylase, which is present in our saliva. So the final breakdown means that the carbohydrates have to be in one of these three forms: glucose, fructose, or galactose. And the mechanisms this happens are with different enzymes that are shown here and also on this slide. This is a really good slide to kind of show you the enzyme and the source of the enzyme and the food stuff that is broken down. So you can see in the mouth, salivary amylase begins the breakdown of starch. Then pancreatic amylase is going to continue that process. Finally, there's brush border enzymes, which are in the small intestine, and those are called dextrinase, glucoamylase, lactase, maltase, and sucrase. And a lot of times these enzymes are named for the chemical that they work on. So proteins are first broken down in the in the stomach, and just like carbohydrates, they begin as large polymers. In this case, they're called polypeptides. They need to be broken down into their building blocks, which are called the amino acid monomers. So this digestion begins in the stomach, where pepsinogen is converted to pepsin, and there's a very acidic pH, which helps to break down those large polymers, those polypeptides. And this chart summarizes the other locations where the protein digestion occurs. So it begins in the stomach via pepsin in the presence of hydrochloric acid. Then there's pancreatic enzymes that also continue this process called trypsin, chymotrypsin, and carboxypeptidase. Finally, there's brush border enzymes, and these three enzymes all have the word peptide in them, so they're easy to recognize. But again, the final result has to be that the building blocks, the monomers, are produced. So the flowchart of digestion then for the fats are shown here. There is a lingual lipase, technically, which is a very minor amount in the saliva. So again, the main molecule that's broken down in the saliva is the carbohydrates with the salivary amylase. There then is gastric lipase as well in the stomach. And that, again, that's minor. So the major breakdown of fats occurs in the small intestine because of the work of both the liver and the pancreas. The liver produces the bile, and that bile then travels to the liver through the hepatopancreatic sphincter. and pancreatic lipases also help to break down those fats. So the mechanism by which this occurs is called emulsification, digestion, and absorption of fats. Fats are a little more tricky because they're more difficult to break down. The first way that they're broken down is through emulsification. And emulsification means that these biosalts, that think of them kind of like detergent molecules, are going to start to break bonds down. It's kind of like the idea of how detergent works in your washing machine to separate the dirt from your clothing. So the next event is digestion, where other enzymes like pancreatic lipases can hydrolyze the triglycerides. Micelles are formed, which consist of fatty acids, monoglycerides, and biosalts. Those then diffuse into the epithelial cells. Chylomicrons are formed, and then those are then transported into the body. And the fat is a little different because it uses the lacteals in the small intestine. That's the one difference between the fats and the carbohydrates and the proteins. So our next slide is showing the digestion and absorption of nucleic acids. This is not as significant, but nevertheless, there still has to be monomers that are used to synthesize the real important molecules that we need for mitosis and then for production of proteins, RNA. So the pancreatic nucleases are going to hydrolyze, use water to break down nucleic acid to those nucleotide monomers. And the enzymes responsible for that, again, are in the pancreas, ribonuclease and deoxyribonuclease. Those should be easy to recognize because they kind of sound like the molecule that they're working on. And then finally, brush border enzymes that are in the small intestine. So the absorption of vitamins, electrolytes, and water occur in the small intestine, and this is fat-soluble vitamins, A, D, E, and K. Then there are water-soluble molecules, vitamins C and B. Vitamin B12, it binds with intrinsic factor. So intrinsic factor is a molecule that's produced in the stomach, and it's necessary in order for the absorption of vitamin B12 to actually occur. And then finally, there's important electrolytes that are absorbed.